Hi, everyone, and welcome to MGFA's What's New in Research webinar. My name is Jenna Umbalo, and I'm the Director of Patient Advocacy and Community Engagement at the MGFA, and I'll be moderating today's session. Before we get started, I'd like to go over a few housekeeping items. Participants will remain on mute throughout the presentation. If you have questions, please submit them in the Q&A box located at the bottom of the Zoom menu, and we'll answer them during the Q&A segments. I'd like to give special thanks to our research series sponsors, Alexion, Argenx, Janssen, UCB, and Immunobiology. And now I'd like to present today's speaker, Dr. Kevin O'Connor. Dr. O'Connor is a tenured associate professor of neurology and immunobiology at Yale School of Medicine. And the major research interests of the O'Connor Lab include human neuroimmunology and autoimmunity. It's anticipated that the studies they are doing will define highly specific treatment targets that will affect clinical improvements for patients with MG. Dr. O'Connor, thanks so much for being here today. I'll hand it over to you.
Great, thanks very much, Jenna. Let me do a screen share here. Okay, should be seeing my screen. Yep.
Yep.
Let me go to presenter mode. Okay. And seeing the cursor?
Yes.
Okay, great. Thanks very much. It's great to be here with all of you. Delighted to speak about some of the work that we've been doing on myasthenia, as well as give an overview of how an academic research lab operates. So I'm going to speak today in two different parts. The first will be a story about where academic research labs fit into the world of medicine and therapeutic development, and how funding is used, importantly, and how we have to compete for funding to keep our labs going. And then in the second part, I'll shift over to some of the research that we've been doing on myasthenia gravis and tell you a story about what we found out about the lasting effects of thymectomy, to give an example of some of the research that we do. So to start, the first part is a little bit about academic research and funding, how it all works. So in general, and I'm speaking in generalizations mostly today, there's lots of different universities that work a little bit differently, but I think this covers what an academic research lab looks like with specific interest in understanding autoimmune diseases and immunology. And so the lab head is called the principal investigator, and that is invariably a faculty member, so somebody at the assistant professor level or above, so assistant, associate, or full professor. And that person, he or she, runs a lab that includes a team of researchers. I put this picture here because this is when we discovered one of the first MuSK monoclonal antibodies that we isolated from patients. And so this is our actual lab, and this was a real event that happened. And here's a more recent picture of some of the group that's been in my lab. And so labs work based on funding. Money needs to be provided to the lab, and I'll talk about that in a little more detail in a moment. But where does the money come from? So it comes primarily from the National Institutes of Health. So if you're at a medical school and you're doing medical research, your funding comes from the NIH. The budget is around $32 billion, and they provide grants for us to do research that we have to compete for to get. Other monies come from private organizations, of course, like the MGFA. And then in some cases, although it's more rare, companies will provide funding for our research as well. Then the other part of this is the university. And so I've put Yale here as an example because I'm speaking specifically about my lab. But what Yale provides in our case is that they provide the lab space. So we work in a building that's owned by Yale. They provide administration, legal, human resources, all of the things that you can think of, lights, heat, core laboratories, which are labs that do functions that serve many different labs. But interestingly, and this is important, not in capital letters, not funding. None of the funding that runs the lab comes directly from the university. It has to be extramural. It has to come from outside sources. So I just wanted to point out what place the academic research lab has in the world of therapeutic development, because the aim is, of course, to help patients. So the mission of the university lab is to understand disease mechanisms, to deeply understand how they start and why they propagate. Sorry, this is my cursor being funny. I'm going to switch to the laser pointer so that doesn't happen. Okay. Jenna, you're seeing my laser pointer circling mission?
Yep, we can see it.
Thanks. Yep. So we're primarily engaged in basic and translational research, basic meaning fundamental understandings of disease mechanisms, and translational means research that can translate to the bedside to help patients. There's a great deal of risk involved in this because many experiments and many hypotheses turn out not to work. Additionally, our function is to train physicians and researchers and faculty members, and to teach and mentor students and researchers. I wanted to highlight this because what we don't do is usually we don't produce therapeutics and we don't test therapeutics. So that's usually left to private industry. They are risk adverse, so they don't like doing research, core research, because it costs a lot of money and the outcome is unsure. But at the same time, there's a huge amount of cost in producing and testing therapeutics, so they take that on where we don't. Generalization of what our place is in this scheme. So a little bit more about the laboratory. What are they made up of? So I mentioned the principal investigator as a faculty member, but who are the folks in the lab? Who are these folks? So they're traditionally trainees, and they're people that are in a particular place in their career, and they are moving from one place to another. So the most senior members are postdoctoral fellows. So these are people that have finished a PhD, and in some cases finished an MD and want to do lab-based research. And after you've finished a PhD, you typically need additional training to become an independent investigator. And so these are the postdoctoral fellows who come to the lab for anywhere between 2 years to 4 years to gain more training and to do research with the PI. There's graduate students that are PhD trainees that are earning their PhDs, and they will stay in the lab usually between 4 and 5 years. And we have a lot of postgraduates. And so postgraduates are individuals who have just finished their bachelor's degrees and are usually going to go on to medical school or to graduate school, and they're taking a gap year. This has become something that's very, very popular now, where students don't go directly from finishing their bachelor's on to grad school. They take a gap year and work with us for a year and sometimes 2. Some labs have professional scientists, people that are permanent employees of the university that work as scientists. These are rare in laboratories, and one of the reasons is they're very expensive. The salaries for professional scientists are quite high. Industry can afford them, but usually academic labs have trouble affording them. So I point out that they're more rare. So for our mission, what do we do? So we're interested in advancing the understanding of disease mechanisms to improve human health. Our other mission is to communicate and share our scientific advances transparently and quickly. And as you can see, I pointed out, we train and develop and mentor young investigators. And I just wanted to mention, as a faculty member, I'm often asked, what do I teach? At medical schools that are primary research institutes, there's a light teaching load for most of us. There's not a great deal of classes to be taught for medical students, and there's not a great deal of classes to be taught for PhD students. They spend time in either the clinic or the laboratory, respectively. And so, for instance, I teach one class, one semester every year, and I have other faculty members help me teach that class. So we don't have a very heavy teaching load. And then I wanted to point out that academic labs are very specialized in most cases. So they often specialize in a specific disease and a specific area of science. And so, for example, my lab, as Jenna mentioned, primarily studies human neuroimmunology. So this is when autoimmunity and your nervous system come together to form a disease. My lab focuses very specifically on myasthenia gravis. And then within myasthenia gravis, we're focused on B cells and autoantibodies. And then digging even deeper, we're looking specifically at the mechanisms used by these B cells to mediate pathology. And we use techniques such as sequencing of B cell specific genes. And this is to say, this is what we do. And it's very rare for an academic lab to switch gears entirely. Like you wouldn't find a lab like mine who studies myasthenia gravis and the immune response switch to genetics or switch to cancer. It's just not usually done because it takes so long to establish a system to study a disease that we usually stick with it for most of our careers. So funding and the budget, how is it spent? So we have to compete for grants. We have to write proposals and request money with these proposals and compete with other investigators to win that money. When we do win, grants are awarded minimally at 1 year, but the longest ones can be 5 years. And so I broke down how we have to spend the money. So one of the biggest expenses for an academic research lab is the salary and the fringe benefits for the people in the lab. So the principal investigator and all of the lab staff are not paid by the university directly. They are paid by the grants that the principal investigator brings into the laboratory. And so in this picture, we have to bring in funding to support the salary of this group of people, all of us, including me. There are some instances where some faculty members have a very heavy teaching load, or if the faculty principal investigator is a clinician, their salaries are usually covered, but otherwise, only for the principal investigator, but otherwise, you have to pay for the entire group of people here. And then the other thing we spend money on are laboratory reagents and consumables. So these are plastics. We use lots and lots of plastics in the lab that are consumable. They can't be reused. And then we have equipment we have to buy. This is a flow cytometer that separates cells based on their type. And we have to not only maintain these, but we have to repair these. They actually belong to the principal investigator, not the university. And so we're responsible for their maintenance and repair. And then, of course, because we're communicating our science, we publish and we go to meetings where we present our data to share our experiences with the scientific community. So publication in scientific journals is actually expensive. Many of the journals are actually owned by for-profit publishers. So publishing a research report in a journal can cost upwards of $4,000 for one article. And going to meetings is expensive. The meetings are a logistical, very complicated logistically. And so it costs money to travel to these meetings and present. So we spend our money there. And then I wanted to mention that the university provides the backbone of our work by providing all the infrastructure, by taking from grants what are called indirect costs. So if you get a grant from the NIH, for example, all of the money goes to the lab to do the research. But the university also gets an indirect amount of money from the NIH to support the infrastructure of the labs. At Yale, that rate is 68%. But for all nonprofits like the MGFA, it's only 10%. And then, so the final slide here, before we go to the next section is, so is it competitive to get grants? It's incredibly competitive to get grants. And so here's a statement from the NIH that they were talking about fiscal year 2019. They received 54,000, approximately 55,000 research program grants in 2019, and 11,000 approximately were funded, and this led to a rate of 20% success rate. So that's the overall NIH. Different institutes at the NIH are competitive at different levels. So one of the most competitive is the National Cancer Institute. There's a lot of investigators competing all for the same pot of money. And so what they're showing you here is the percentile for your grant, meaning where did your grant score? And so if you score below 10%, meaning you beat 90% of the other grants in the score, you've got a 50% chance of getting funded. If you slide down to 7%, if you beat 93% of the other investigators for scores, you're close to 100%. So it's really competitive. The grants that are submitted to the NIH are usually by established investigators. And I actually sit on the review committee for one of the NIH committees, and they're all good. And so this line that separates getting paid and not getting paid to run your lab is very fine, and it's very, very hard to do. So it's competitive, but with that said too, competition makes sure that everybody's working very hard to put forward the best science that's going to help patients in the long run. So it's a good thing that it's hard to get this funding. So that's an overview of what it's like to run a lab and where we stand in the development of therapeutics. And I just wanted to say one quick thing on the last slide here. Does it all work? And it's not a perfect system, but it does work. And I just wanted to show two quick things. This is from the National Library of Medicine that keeps track of publications from research labs where they're communicating their science. And in 2017, where I took this page from their website, there were over 800,000 publications. So the NIH is getting something for their money. And just to point out something specific for myasthenia, a subject we're all interested in, is here are two papers that are communications of science that were reported by a laboratory run by Angela Vincent, who is a outstanding scientist, has made major discoveries in myasthenia. Both of these are from Angela Vincent's lab. And in this first one, she demonstrated that patients that were labeled as seronegative, so seronegative myasthenia gravis, many of them actually had antibodies to the acetylcholine receptor, meaning that they were previously undetectable, but she developed a system for detecting them. And this is a big deal for patients with seronegative myasthenia because it gives their clinicians an opportunity to understand how to treat them better. And then another landmark study from Angela's lab was that she discovered MuSK myasthenia gravis. So she was again looking at patients that didn't have antibodies to the acetylcholine receptor, and she found out that many of these patients have autoantibodies that bind MuSK. And so this paper, published in the prestigious journal Nature, showed that there are patients with myasthenia that have MuSK. So this is a product of academic research labs. And I think you can agree that it's pretty important, and there's been some success here. So I'm going to shift to part 2, which is about some research that my lab has done. But if Jenna had some questions that came up in the Q&A, I can take them because we're really switching subjects now.
Thanks so much. Yeah, we do have a question from outside of the Q&A. The first one is, what happens when a lab doesn't get funding, or say when the NIH cuts funding?
So the NIH does continue to cut funding from time to time, and even if you win a grant, sometimes they trim it, they scale it back, and you just have to make do. If there are labs that fail to continue to compete for funding and simply run out of money, and that goes on for a long time, typically what happens is that lab will close.
Okay. And then this other one is, during COVID, we've had a lot of virtual conferences that must save money, but what, if there are any, are the downsides of doing scientific meetings virtually?
Oh, that's a good question. Thanks for asking that. They've been cheaper. The meetings have been cheaper because you don't have to travel. But the registration for the meetings has actually not gone down very much. And I guess that's because of all the logistics of putting together a meeting virtually. So the downside is that you don't get to sit with your colleagues and spontaneously start discussing things that you would find interesting, and make initial discoveries together by having discussions about your research. Zoom is not a natural format for people to keep communicating. So all of the struggles we all face with Zoom in our daily lives, whether it's parent-teacher conferences or or scientific meetings, it's all the same in that it's valuable and useful now to keep us going, but it takes away the human dynamic of sitting down with somebody and having a simple chat over a cup of coffee. Let me add one more thing to that, Jenna, but the other good thing about it is that there's some efficiency. So rather than traveling to, I live on the East Coast, so to the West Coast for a meeting that I'll attend for a day and a half, I would lose a total of 3 days because of travel. So you can go to a meeting for a half hour and still be down at the dinner table by 5 o'clock at home. So I guess that's been some advantage.
Yeah, that's a good point. So we got a couple more questions. The first one is, as a seronegative GMG patient, can I volunteer my blood to be studied for antibody discovery?
I think the answer to that is an absolute yes. So at Yale, the Myasthenia Gravis clinic at Yale is run by my colleague, Richard Nowak. Dr. Nowak is a neurologist. And when he sees patients in his clinic, he invites them to participate in the research studies that our lab is doing. And I think that's the case for many neurologists that are working at universities. Jeff Guptill at Duke does that. And so I think that you could speak with your neurologist and ask if you're able to provide some specimens for our research. And it may be research at the local university where you live, but it's done at many places around the country.
That's great. It's great to know. This next question is, have you had any success with private foundation funding? And if so, which ones are particularly interested in autoimmune disease?
I assume this means private foundations like the MGFA. Do you think?
I think so.
Okay, so sure.
Yeah.
Go ahead, Jenna.
I was going to say, maybe because you were talking about NIH funding earlier.
Right. So, yes, private foundations are very valuable in providing funding for research labs. And I'll point this out. I need to use the example of the MGFA. So the NIH grants, as I was pointing out, are very, very competitive. The MGFA grants are competitive also, but they're focused, meaning that if you work on myasthenia gravis, then applying for an MGFA grant, you're within a group of individuals that are also applying for that grant as well. And I think I still say it's competitive, but there's fewer people asking for the same pot of money. So you have a good chance if you apply a couple times to be successful in getting some work supported by a group like the MGFA. And I'll just say this, Sam and I were talking about this, Sam Matheson, the CEO of the MGFA. It's critical for smaller organizations like the MGFA to provide support for labs because the system requires that you have some data to apply for a grant. And what I mean by that is you can't apply for a grant and propose an idea without having any data to support your idea. And that's especially true of the NIH, where the grants can be upwards of $2-3 million for over 5 years. That's a really big grant to sustain your lab for a long period of time so you can do research without interruption. In my case, the MGFA grant that I was awarded a few years back gave us the opportunity to get started on some MG research that we were interested in and build the preliminary data that was necessary for us to apply to the NIH and win an award for, I believe it was $1.7 million. And it sustained my lab for 6 years straight without interruption. So yes, we've had success, and it's a very important part of the funding support mechanism.
That's great, thank you. That's it for the questions we have for this section, so we can move on.
Okay, so in part 2 here, I'd just like to tell you a little bit about some research that my lab has been doing and demonstrate what I mean by translational research, that which we do that translates to directly helping patients. And so many of you in this audience know that pathogenic autoantibodies cause this disease. So your body makes autoantibodies that bind to the acetylcholine receptor at the neuromuscular junction and interrupt the transmission between the nerve terminal and the muscle fiber. And so autoantibodies are made by B cells, and there's 3 types of B cells, and it's important to understand a little bit of the differences of them. And I think we've heard a lot about B cells in the news because of COVID, and I wanted to just point out that there's 3 different types. There are naive B cells, and so these are B cells waiting to see something new. So COVID's new, and all of us had naive B cells waiting to bind to COVID. They were ready to react. When they do recognize COVID, they form memory B cells, so you don't get the virus again once you've had it or a vaccine. And then the 3rd type of B cell is one that makes antibodies. And so these are the types of B cells that secrete these antibodies that you hear about in the news regarding COVID. So they're the circulating antibodies in your blood. So in a normal B cell response, when you have an immune response to anything, B cells are born in your bone marrow, and then they circulate in your blood. And then they might encounter an antigen, and this could be flu or a flu vaccine, or COVID or the COVID vaccine. And then in your lymph nodes, what happens is these B cells react and produce what I showed you, the memory B cells. And then they produce B cells that make antibodies as well. So it makes a memory response so that you can react again quickly if you get the same infection. And then they make B cells that produce antibody to protect you from the virus that may have infected you at this time. And then you produce a set of long-lived B cells that keep making this antibody. That'll help get rid of the vaccine or get rid of the virus, or respond to the vaccine so you're ready to get rid of the virus. And so the immune response in AChR myasthenia looks very similar. This is the same picture, just superimposed with one additional component that I want to talk about today, the thymus. So in some patients with early onset myasthenia gravis, the thymus is involved. Thymus is not involved in some other types of myasthenia, but in the early onset type, many patients have what's called thymic hyperplasia. And the diagram is the same. There's B cells that produce autoantibodies. And what we wanted to do was find some more granularity here to understand this a bit more. And we were focusing on the thymus. And I'll tell you why. It's well known that in the thymus tissue of patients with myasthenia, there are B cells. And there are B cells that make autoantibodies to acetylcholine receptor. So this is a study back in 1978, where they took thymus tissue from patients that had thymectomy, and they put the cells from the tissue into culture, which is just like a soup that allows cells to stay alive. And they found out that if you left it for a few days, you would get antibodies to acetylcholine receptor if the thymus was removed from a patient with myasthenia gravis. So what that says is that we know that the thymus in patients with thymic hyperplasia, that subtype of myasthenia gravis, they have B cells in their thymus that make the autoantibodies that cause the disease. So removing the thymus, having a thymectomy, has been a treatment, long-standing treatment for myasthenia. And recently, Gil Wolfe and Henry Kaminsky and others published a trial in the New England Journal of Medicine where they demonstrated that thymectomy is helpful for patients with myasthenia gravis. So they're showing you here patients that were on prednisone alone, and then patients here that had a thymectomy plus prednisone. And they're looking at their QMG score on the y-axis. And so the patients with the thymectomy over 36 months, 3 years, on average did better than those that were just on prednisone. So there's some evidence that overall, that thymectomy is beneficial in patients with myasthenia gravis. So we asked a question about what happens to patients afterwards. And so I want to point out why. it's clear here from this diagram that the MGFA, sorry, this is a QMG score. So this is a disease score, like how burdensome is the disease. It doesn't go to 0. So thymectomy is not a cure. The patients still have myasthenia after the thymectomy, and they still demonstrate symptoms of the disease. They get better, but not completely. So we asked a question following this diagram again about what happens when you do a thymectomy, focusing on B cells. And we asked, if you take the thymus out, and therefore you take out these B cells that produce autoantibody that cause the disease, but patients don't get completely better. So we asked, are there still B cells in the periphery, in the blood, after the thymectomy that make AChR autoantibodies? And are they derived from the thymus? And then we asked, if they do remain, if you have more of them or less of them, does it translate to the clinical outcome? And so we investigated this question. And here's the way we did it. We partnered with the MGTX trial, which was a trial where they were testing whether thymectomy was beneficial. And during the time of thymectomy, patients had their thymus removed and they had some blood drawn. And then a year afterwards, they had their blood drawn again. And we have these samples. We were provided with these samples in my laboratory. And we asked the question that I proposed to you. And we used we took the different specimens at 2 different time points in the thymus tissue, and we used a particular type of sophisticated sequencing that allows us to identify B cells in the blood at time 0, blood at 12 months, or blood in, or B cells in the tissue. And we asked if any of those B cells were clonally related. So B cells, if you were to get a COVID vaccine, you would make B cells that would produce antibody to the virus coat protein. You've heard about this protein, the binding protein that is the target of your antibodies. And so a collection of B cells will make those antibodies, but these B cells are clonally related, meaning one B cell starts to react, and then it produces a bunch of daughter cells that are identical clones of it. So it just keeps making more of them. So you can find these clones in these different samples. And the way we identify them is really with a barcode. So each B cell has an identifier that's unique to the B cell. And every daughter that B cell produces has that same barcode or clonal identifier. So we can trace these B cells over time and in different tissue compartments. And what we found, here's the data, and I put it in a Venn diagram. And on the left here, you have the blood at time 0 when the thymus came out. And over here, you have the thymus tissue. And over here, you have the blood at 12 months. And these are the total number of B cells that we measured in each sample. And then where they overlap is where we found the clones that were identical in the different compartments. So meaning that this set of B cells is 553 B cells were present in the blood at time 0. They were present in the thymus at time 0. And then they were still present in the blood at time 12 months later. In this example, I chose 6 months. We had one patient that was 6 months. But the point is, the B cells have expanded clonally, and they can be found to persist in the blood after the thymectomy. And so we did some more analyses, and we asked about these B cell clones that form trees. So B cells, when they clonally expand, it looks just like a family tree, like an ancestry tree, where there's the parent B cell, and then there are generations that keep dividing afterwards. So sons and daughters, mothers and fathers, and they keep going. And then when we set up a family tree of a B cell clone like this, we asked, were the early ancestors mostly found in the blood or the thymus? And you can see here, I've colored in blue, that they actually are found in the thymus. So our data suggested that the early ancestors of these B cells started in the thymus and then emigrated out into the blood during the thymectomy, at the time of thymectomy, and then were present months later after the thymectomy. So what that means is this: if you think about this diagram again of a normal B cell response, we know that there are B cells present in the thymus that produce autoantibodies that target AChR and cause this disease. We now know that B cells that are found in the periphery, that means in the circulation in the blood, that they're derived from these clones that are in the thymus. And perhaps they're even set up to be long-lived cells that continue to produce antibody, much the way your B cells will produce the antibodies after you get a vaccination. If you take the thymus out, you're taking out these original source of B cells, but those that are present in the blood, and perhaps those that persist in the bone marrow that continue to produce antibody, are the reason why patients that have a thymectomy get better, but the disease is not gone because the B cells that were born in the thymus and then emigrated out remain. And so are these clones derived from the thymus? Our data suggests the answer is yes. And do they remain in the periphery or the blood after thymectomy? And our answer is yes. And so looping this work that we did back to my first part of the discussion about academic research labs, what's the product of this research? So, well, it's everything that I mentioned in the first part. Training individuals who are going on to careers in science and medicine. So this work was headed up in my lab by a young gentleman by the name of Roy Chang, who has just finished both an MD and a PhD at Yale, and he's gone on to finishing his medical residencies now. And Min Pham is a PhD student in my laboratory who just recently started and participated in this work too. So there's the trainees. And here's the publication. So we published this study in the Proceedings of the National Academy of Sciences. And so we communicated these studies to our colleagues to suggest to them that when treating patients with thymectomy, physicians and clinicians and surgeons know that the disease often continues while patients can get better. It does continue. And I think our data suggests we have an understanding now why. And it's because the B cells that are clearly present in the thymus, while they are removed during the thymectomy, their progeny remain persistent within the individuals after the procedure and may contribute to the lasting disease that's observed. And so finally, I just want to end by acknowledging the people that helped do that work. So I mentioned Roy and Min. Casey Lee was another trainee. He was a post-bac who finished his bachelor's degree and then did a gap year, and now he's getting an MD PhD at Penn. Another principal investigator collaborated with me, Steve Kleinstein, who has a postdoc named Ken, who did a lot of the computational analysis. Dr. Nowak is the neurologist who sees these patients, who connects with us to do that work. And Frank Detterbeck and Robert Homer are surgeon and pathologist, respectively, who helped us acquire the thymus tissue and the blood. And then we also collaborated in this work with Henry Kaminsky, who ran the MGTX trial. He's at George Washington. And Angela Vincent, the famous MG scientist who's at Oxford, worked with us on this project as well. And Chichi Aban, who's a statistician at the University of Alabama, helped as well. And this is all the funding that we had to do this work. So the NIH supported some of this, the MGFA did. Very fortunate to have funding from the MDA at this time, Conquer MG, as well as the newly formed MGNet. So with that, I will I'll end and move on to take questions from that next part.
Thanks so much. Thanks for sharing your findings. That was really interesting and really great just to see the journey from discovery to translation into science and sharing the information with the community. So the first question is there any way to attack the specific B cell that is still present after a thymectomy, the daughter B cells? Since you can track the B cells with the identifying barcode, can you specifically kill off that B cell?
So that's a great question. Thanks for asking it. So the answer, there's a two-part answer here. So the answer is yes. There are therapeutics. I just want to say at the onset too that I have a PhD in biochemistry. I am not a physician. I'm not a neurologist. So I'm only speaking from the laboratory side of things. But yes, there are therapeutics that target B cells in the periphery. And so you've probably heard, many of you in this field and patients and physicians have heard about rituximab. So that's a drug that will specifically target B cells and eliminate them. So there are therapeutics available that can target the B cells that are persistent afterwards. And I think that in treating MG, that many neurologists do consider what to do next after thymectomy to further benefit the patient by going after the additional immune response that's present. The second part of the answer is that the question also alluded to whether or not you could target the specific B cells. So that is exactly where the next step of personalized medicine would like to go. So wiping out all the B cells will include wiping out those that are producing the autoantibodies. While that's good, the bad side of that coin is that you're wiping out all the B cells. So you don't necessarily want to do that. The better way to do it is only attack the B cells that are making autoantibodies that target AChR or MuSK. And the therapeutics are not quite there yet, but it alludes to the type of research that we're doing. We're trying to find how to identify very specifically only those B cells that produce AChR autoantibodies so they can be targeted.
Thank you. So this next question, I think it might be along the same lines, just with, can anything be done to the blood to remove the errant B cells?
I think the same answer does apply. So rituximab does work at eliminating B cells that are circulating. It also eliminates B cells in tissue, so in lymph nodes and thymus as well. But yes, I think that targeting them with rituximab, but many of the other therapeutics that are available to treat MG also diminish your immune response as well. So even prednisone is effective at that.
Okay, thank you. This next question is, if these B cells are still persisting after a thymectomy, is it worth getting the thymectomy?
Not a clinician, so I don't know if I can answer that and provide medical advice, but from the immunologist point of view, you're taking out a huge source of autoantibody producing B cells and T cells that are helping them. And it's clear from the literature that the clinical trial demonstrates that a thymectomy is effective in helping patients with MG. So from the immunologist perspective, I would say yes.
Thank you. Okay, this next question, can you see this question on the
I
chat?
can't see any questions.
Okay. Because it might be,
Oh wait, I didn't have the Q&A open. Sorry, now I have it. Which one are we looking at? Open, okay. Have you seen any MG treatments linked to this one?
yes.
Yes, yeah. Have you seen any MG treatments linked with treating post-COVID symptoms, dealing with both, unfortunately, neurologists, but things for rituximab really help? Not a question that I can answer. Have to speak with your physician for that one. Sorry.
That's okay, thank you. And then we have another question, which is, how does having these B cells persisting in the blood after a thymectomy affect a patient with MG?
Well, if they're still there and they're still producing autoantibodies. So the autoantibodies when they migrate to the neuromuscular junction, are causing the disease symptoms. So if the B cells after thymectomy that produce those autoantibodies persist, then they will continue to produce autoantibodies, and you'll still have manifestations of the disease.
Thank you. How quickly do the B cells reproduce, the remaining cells after a thymectomy? Do they continue to reproduce?
Yeah, that's a really good question. We don't really know the answer to that. Sometimes after thymectomy, some patients will get much better, and some patients don't respond well at all. So I think that it varies based on individuals. And so we don't know why in some cases those B cells will rapidly reproduce, and in other cases those B cells will remain quiet. We're not quite sure. But these are the types of questions that we're keen to answer in my lab and the type of research that we do.
Thank you. This next question is, do seronegative GMG patients respond to thymectomy or B cell depletion?
So part of the answer is a neurologist answer that I can't get into, but I can say this. So I think there are a subset of seronegative patients that may have autoantibodies to AChR. And Angela Vincent, as I showed you in the slide at the end of the first part, published a paper demonstrating that there can be AChR autoantibodies in seronegative. So I think a subset of patients probably would, and this is just based on the immunology, I think a subset of patients with seronegative MG might benefit from thymectomy, yes.
Thank you. And then we have a comment here that's, thank you for all the research you're doing to move the understanding of MG forward.
Well, thank you. We're very happy to do it. Thank you.
Well, this concludes the questions that we have. So Dr. O'Connor, on behalf of the MGFA, I want to thank you so much for your time today and for educating us. I want to thank everybody for your time for participating, and encourage you to be on the lookout for future research series webinars as well.
Thanks, Jenna. Thanks everybody for joining. It was great to be here today. Thanks again.
Thank you.
Okay, bye-bye.
Bye.